Hi everyone, I'm Apurva and I'm a CFP intern based at Q with Mohammed and Laura Kelly. So I've been working on this internship project for the past few months on exploring the genome of an ash tree that is less susceptible to ash dieback. So firstly, I would like to thank DEFRA and CFP and Forest Research for the funding, and also my managers and supervisors, Mohammed, Laura, and Daniel, and Richard Bugs, and also the whole plant health and adaptation team at Q, and mainly also Apocrita at Queen Mary for their high performance computing resources. So, so I'm sure you've already heard Nathan's talk and have a introduction on ash dieback. So Fraxinus excelsior, which is European ash, is an important native species in the UK, and it is under severe threat due to ash dieback and another caused by this fungus Hymenospace fraxinus. And we can see that from the data, 12% of the broadleaf woodland in Great Britain is ash, and it is it is an important species which is under threat. And based on the data from Forestry Commission, we can see how the disease has spread over the years. From this is on my left is the data from January 2015 to how the disease has spread to September 2023, which is quite rapidly spread. And here in some of the pictures, we can see how some of the symptoms of the disease of leaf blackening and the crown dieback, and especially here, we can see how the stem of a tree is rotten inside due to the ash dieback. So our research team at Cube uses genomics to study ash trees and ash dieback. So this was the first paper published in Nature in 2016, which published the first reference genome, is used as a which provided us with the foundational genomic resources for further research aimed at improving our understanding of susceptibility to major threats to ash and for providing tools to help tackle these. And then there was another paper about the genomic wide association studies to identify loci associated with low susceptibility to ash dieback. And also another paper about comparative genomics analysis to identify candidate genes for low susceptibility and defense against emerald ash borer. So as we can see, based on our research, we come to one conclusion that a good reference genome is crucial for the subsequent studies to take place, like genome association studies or genome environmental association studies, or to come up with a linkage map or functional annotation. So a reference genome is used as a genome which is assembled and used as a baseline standard for comparing and studying further sequences. So the first reference genome that was published in the 2017 paper was based on a susceptible individual. So for complementary studies in our group, there was another individual which was sequenced based on PacBio Hi-Fi platform, which we are, for simplicity's sake, we are calling PG5, and it was found to be low susceptibility to ash dieback. So my project mainly aims at annotating this genome, and based on the data that we have sequenced, the long-read RNA-seq data that we've sequenced from different tissues of this individual tree, and also augment this data with some of the other short-read RNA-seq data from the original ASH reference genome we had to improve the genome annotation at the end. So genome annotation mainly starts off after you sequence the genome, you assemble the genome, whereas when you sequence, the data is given off in thousands of fragments of the sequences. So we need to assemble them into one huge sequence. And so this assembly was done by another postdoc in our team previously, and now I'm trying to annotate this, and annotation mainly deals with what genes are present, where they are, and what is their role. So this is the pipeline that I've come up for the annotation, and it mainly consists of going through quality checks initially and then filtering out if there are any adapters or contaminants present, and then mapping it to the reference genome and also masking it and doing gene prediction and functional annotation, and finally doing quality assessment. So there are different software tools that I have used along for each of these steps, and there are different steps for along this process, and these are some of the softwares I've used for any who may be interested. So, because we've collected three different tissue samples for our long read data based on Oxford Nanopore technology, and there were duplicates for each of the tissues. These were the number of sequence distributions across the different tissues, and the number of sequences were varying. And then this is for the short read data, which was based on Illumina platform for different tissues of cambium, flower root, and leaf. And then after getting these statistics, I've done mapping, so using Minimap2 and HISAT, and we had very good mapping percentage of more than 94% across all the tissues for long read and 85% for short read. And then a Breaker 3 tool was used for gene prediction, and this had results comparatively for a previous individual which was studied in the lab had 35,008 genes, and in the current individual that I'm analyzing had 36,700 genes approximately. So this is where my work is, and currently I'm trying to do the functional annotation of genes using NTAP and GFAX tools, and we further work is planned to identify the genes in this individual that are missing from the original reference genome, and further work is planned to identify if any of those genes are responsible for giving it low susceptibility to ASH dieback. And finally, I would like to talk a little bit about my learnings from the internship, and it was amazing experience, I would say. And I have learned that working with real data and real-time research is not easy, and it's all about learning new things and unlearning sometimes, because you might go in thinking, I know this process, but it's totally opposite of that. And I think it's okay to not know things, especially when you're starting off, and being adaptive to learn is more important. And sometimes you'll have to keep going back to the previous step to make sure you're going in the right path, and it's not always just moving forward. So just be okay with it and you'll be fine. And things take a lot of time and practice to get that done, what it looks on a paper when you read. So yes, things take a lot of time and you cannot get it all done and right in the first time. So I think the only way you can keep pushing forward is being humble about learning things and curious and consistent with your work. And I've learned a lot of science and other things from my supervisors, my colleagues, my peers, and my managers. And along the way, I also made some amazing friends and memories. So it was a great experience. And thank you, Forest Research, for this opportunity and for the opportunity to talk at this webinar series. And thank you, everyone, for your time and attention.